Good. So I have elected to give a more mathematical talk than most here. So I'll talk about catalyzation. I will talk about a problem we have done, we being Dylan Sislow and myself, on constructing Waddington landscapes with stem cell differentiation. And then I will talk about a more challenging problem, which we're partway through, to look at the mouse blastocyst, a situation where there are many cells, there are internally generated morphogens, and of course, there's also real morphogenesis. Okay, so the phenomenon of canalization begins with this book from Bateson from the 19th century, which is basically a collection of 500 pages of these so-called homeotic transformations, where a leg goes to a wing, or as you see there, a thumb to more fingers. And you will note that he coined the term homeotic, and Victorian scientists were well versed in Greek, it seems. Another example of canalization is this famous Mangold-Spemann experiment, in which you, in which you transplant a bit of tissue from the dorsal lip of a stage-matched embryo. You put it on the ventral side of another embryo, paste it in, and what you will see, if you have good hands, is the figure on the lower right, in which you have a replicated a trunk axis, which demonstrates induction because it is the host tissue that is induced by the transplanted fragment, dorsal lip, to produce a second axis. More interesting are these regeneration systems, salamander and fly. On the upper right, you see what was done was to wound the salamander limb on the anterior side to bathe the limb in a solution of the posterior morphogen, and the opposition of those two morphogens then gives you this outgrowth shown in green with rather high fidelity. I recommend to your attention the problem on the lower right, which is an old paper from Meinhardt, who showed how in a disk you could use three morphogens to select the center of the disk, which will then be the distal tip of the fly leg as it grows out. And there again is the opposition of two morphogens, in the fly case, wingless and DPP, which when you put them in clones, you can get this duplicated distal tip, which you see on the left for the fly, which is rather reproducible. And again, this phenomena is very interesting from those who are interested in morphogenesis and is not well studied. Okay, so I have here the cliche image of Waddington's landscape. And what I would like to do is to ask how quantitatively we can justify that. So you all know that if I write down a bunch of Michaelis-Menten equations, they are indecipherable. Just as quantum chemistry product wave functions are indecipherable, one wants something much more akin to Pauling's theory of the chemical bond. And mathematics deals with these problems by defining categories of problems and enumerates the types of solutions, which I will show you in this context. Another example being actually the stability of planetary motions, which is a separate story. And this quip from Dennis Sullivan is actually very insightful, although it seems frivolous. Okay, so the relevant phenomenology here, or the mathematics, proceeds by taking flows on the left, i.e. arrows, and converting it into landscapes, i.e. topographies on the right. So you see, for example, this doesn't work at all. You see, not work. Okay, anyway, you see a green circle, which is the top of the landscape. From the green circle emanate arrows. The arrows eventually collect at the red circle on the right, which is the sink, and will sometimes pass through the saddle point X on the left. And obviously, there's a topographic version of that scenario. So we want to equivalence flows and shapes. That's how the mathematics works. So a glove has 5 attractors, which are my fingers, 4 saddles between the fingers, and 1 repeller of the wrist. And you could all visualize how to distort that shape, the manifold, and turn it into a sphere, i.e. the North Pole is the repeller, the South Pole is the attractor, and the 2 are topologically equivalent. So Smale, in the late 60s, proved the so-called Poincare conjecture in dimensions bigger than 5. The proofs in dimensions 4 and 3 got 2 other individuals Fields Awards, and we borrow that technology from Smale. Okay, so I then enunciate a theorem to you due to Smale, is that life is all downhill, which you perhaps know intuitively ever since you were a zygote. So one defines, as I alluded to, a type of system. So Morse Smale systems have a discrete number of rest points, i.e. saddles, attractors, sinks, or orbits, no chaos. The assumption is made of structural stability. That's a very potent assumption in mathematical terminology. It's very much akin to stating that embryonic development is robust, i.e. you kick the embryo, you jiggle the embryo, you still get a fetus and an adult out the other end. If you put aside if you assume there are no orbits, then Smale showed with some technical assumptions that the flow equals a metric times the gradient of potential on a Riemann manifold. That, and note there's an equality sign there, not sort of like, maybe like, or somewhat like equals, right? So the flow builds the metric and the potential. The metric means that that there's a notion of downhill, but as a good skier, you zig and zag. When you allow periodic orbits in the Morse Smale system, it's of course no longer a gradient system, but there is still a notion of Lyapunov exponent, i.e. there's a notion of downhill, even though the flow is not the gradient of anything. So in this sense, I would say a Morse Smale system is like a Waddington landscape. Obviously, the mathematics is done for deterministic systems, not for real biological systems. And of course, whether this applies to real biological systems is a matter of fitting data and parameters. So obviously, we could ask, how do landscapes change with parameters, i.e. bifurcations? How do the bifurcations fit together, which is the global parameter space? And then there's a short list of geometries that we expect to see. And the goal, of course, is principled fits to data. Another interesting theorem is that there are 2 bifurcations that suffice to converse Morse Smale systems on the same manifold. The first one is just what's called in the trade saddle node. It's the glove to a sphere. So that you all have seen before. The other one, which you have also implicitly seen but not perhaps articulated, is this heterochronic flip. That is termed a global bifurcation because it changes the connectedness between the various rest points. So in particular, the saddle point in little a at the top, which separates the attractor capital A and then the other 2 terminal states, B and C, the connectedness between the saddle little a and B and C changes. And that change, which you could imagine happening because of feedbacks and whatnot in the system, which I will elaborate in a bit, is the change in the connectedness of these insets and outsets, which is called in the trade a global bifurcation. Of course, it very much looks like a stem cell going to 2 possible terminal cells. Now, it is mathematical that these 2 bifurcations are both codimension 1, i.e. if you have an n-dimensional parameter space, these bifurcations will occur on n-1 dimensional surfaces. That's theorem, which is trivial to show. And therefore, unless there is parameter tuning, these are the ones we expect to see. That's these are the ones that we expect to see. That's a very strong statement. Of course, all true for deterministic systems. Now, you obviously know some exceptions to that, such as the pitchfork bifurcation, but these all require symmetry, i.e. the pitchfork requires, in the magnetic context, a zero field and a transition temperature. And similarly, work of Corson and company showed that in the fly NOTAM, the long-range notch delta repression can also give rise to more complicated bifurcation diagrams, but what one was patterning a naive field of cells, which is a non-generic state. Okay, so let's talk about two applications of these ideas that I will elaborate a bit on to see whether they're actually useful for dealing with biological data. Okay, so the first application published in PNAS a year ago, of which Dylan Sislow, who is here, is sitting somewhere in the back if he's not out paddleboarding, was published in PNAS. So the system is one that the Briscoe lab has worked on for many, many years and know very well. They start with stem cells. They convert them to progenitor cells. And these progenitor cells, as you see on the upper right diagram, then are exposed to varying amounts of sonic hedgehog, or SAG, from the floor plate and acquire different fates. And these fates, because the authors have studied these systems in the embryo for many, many years, they know how to choose antibodies to proteins such that in combination, they could identify all the states involved. So combinations of these 5 factors will identify states. And that is all anchored in vivo data. So what we do with this material, with this measurements in 5-dimensional space, is that we look at the union of the data, all points, that we use to define a so-called base potential, which, among other things, its points of high density are points of low potential by the usual expressions, and correspond to the actual states in the system. As a function of, the experimenters then add sonic hedgehog to the cells at varying concentrations and for varying durations, and measure as a function of time what the cells do as regards to in this 5-dimensional space. So as a function of sonic hedgehog, time and level, we then define an interpolating potential, which we use to, so the fitting parameter, the fitting process is that we take the potential height at the various states corresponding to cell states, and we allow ourselves to move those things up and down in energy, and we then drape a smooth potential across those points. And that then defines the sum of the potentials, then defines a path integral, and the path integral is then used to fit time-dependent data as to how the cells slide down the surface. We find that the cell states are locally stable as regards the potential. So you simply take the potential, look at the second derivative, and you find that the eigenvalues are negative, i.e. it's a stable state. These stable states are connected by one-dimensional saddles, i.e. saddles that are contracting in 4 dimensions and expanding in 1 dimension. That's a result, not obvious. And so consequences of this fitting, and again, this is all published, so you could look at the details, which I won't go into, is that the actual data points construct a one-dimensional manifold living in a five-dimensional space. And the flow contracts onto the manifold as it's also sliding down the manifold. With this construction and that fit potential, we then can fit the Kolmogorov decayal divergence of the data as it slides down as a function of time quite accurately. This allows us to then, having fit some time course data, allows us to predict some arbitrary time course data. This phenomena, this fitting procedure also has the consequence that it allows us to interpolate potentials, i.e., if we have morphogens 1 and 3 that bound the value of morphogen 2, we can interpolate and find the potential 2, which interpolates between the potential 1 and the potential 3. Now, the virtue of that is that, you have non-interacting energies that are added together in the exponent to predict gene epistasis. That is non-trivial, and it comes about sort of for free from all these geometric models. We obviously can use this to apply to many stem cell differentiation problems, and it's a very tight but non-trivial construction, which Dylan did with very little input from me. Okay, so the other problem I want to briefly allude to is the mouse blastocyst, which is a situation with many cells and internally generated morphogens and also morphogenesis. So here, the so-called intercell mass on the left undergoes a lineage bifurcation into an epiblast, which becomes fetus, and primitive endoderm, which is extraembryonic. That's, if I may say so, the Ising model of lineage bifurcations. It's a regulative system, which means that if you take one embryo or half an embryo, or two embryos or half an embryo, or you mix together some block cells that cannot assume one fate, the remaining cells, as accurately as possible, assume the missing fate. So the system is very tightly locked into a fixed ratio. We know in great detail the morphogen that controls this signaling. The epiblast makes FGF. FGF inhibits the epiblast, and the FGF encourages PE. Okay, there are two ways to perhaps fit this data geometrically. One is this heteroclinic flip, to which I introduced earlier, for which the ICM state at the top becomes either epiblast or PE. The alternative is a butterfly transition on the lower left, which is topologically one-dimensional because it's three fixed points and two saddles on a line, topologically line. So with Arksham and Raju some years ago, we produced a simple model for this transition, which is just the simplest polynomials for the landscape, uniform FGF over the embryo, which tilts the landscape favoring the two final states. FGF is made by the epiblast cells in some simple polynomial way, and this fits all existing experiments. Now, the open issues, which are many, require us to do our own experiments. So if you ask a biologist, how does an embryo measure time, you will get various mumbles about days after mating, cell number, and sensitivity signals. However, I would maintain via the Waddington analogy that time is really a system property, i.e. is how far down you are on the Waddington Valley, which is a fitting parameter. And furthermore, if you imagine that there aren't Waddington Valleys, but there are Waddington Plains or something, some higher dimensional thing, I submit that you would be very challenging to imagine how metazoans are here to worry about the problem. I don't think we would be here. We want to also look at partial penetrance, and we want to look at commitment, which means what is a cell really stuck in a valley? And we want to probe, we want to kick the cells when they're in valleys to actually see that the valleys are there. So we do this. So we have mice that are doubly labeled for an epigene and a PRE gene. We have dual light sheet illumination, so we could run for almost 48 hours with 15-minute resolution. We can find, we being they, can find segment all nuclei, register, and track all cells. And we could fiddle with it with external drugs that will tilt the fate plane to either one fate or the other. There are controls you have to do, which have been done. The issues that we realized when you actually are confronted with data is how do you go from actual embryos to some intrinsic and common space that you think is common to the process. So you have to, as I will show you, one intrinsic space has to fit all experiments, but you have to fiddle embryo by embryo the fluorescence levels, and you also have to fit the developmental time, and that's essential. So this is a sample of how cells in fluorescence space move around. So they initially were clustered together, and at the end of the movie, you have an epi cluster on the left in red and a blue PRE cluster on the right, lower right. Many different fluorescence ranges have to map into the same intrinsic space. So on the left, I show you in fluorescence space multiple snapshots of the time evolution of embryos in fluorescence, and you see the numbers are all over the place. One has to normalize those numbers to get the pictures on the right in the intrinsic space, which are sitting at 1, 0, and 0, 1 by convention. And one has to, so embryo by embryo, you could fiddle the fluorescence mapping and the initial time, but then you have to map into an invariant space. So this is an example of, on the left, the fluorescence space, and on the right, the intrinsic space with the mappings between them, which are invertible and one-to-one. And you see that things start in the lower left and then move and diverge to the lower right. And you could ask, to what extent is there a common intrinsic space to which everything conforms? And here's another example, which is not crucial. Okay, so so we're in the middle of doing all this. We have these technical means well in hand, and we are now fitting multiple embryos. So to conclude all this, we have to acknowledge that genetics gives us the parts list, genes, but a very myopic view on how to actually fit a model. If you want to fit a model, the most useful data is not a clean endpoint, as is the case for a screen, but actually dynamic interventions that allow you to perturb the thing when you're going in and among the saddles. And that requires a model both to interpret and design experiments. So it's really a joint theory and experimental task. Collaborators on this work over the past 5 or 6 years are multiple and, as you could see, sort of widely dispersed now on the theory side and experimental side. Currently, Sislo and Woon Her are with me in New York. I note two slightly frivolous things. One, I have a word of encouragement from Charles Darwin in a letter to Wallace, who says, I'm a firm believer that without speculation, there's no good in original observation, coming from Darwin himself. I also want to flag for you this absolutely hilarious one-page article by Graham Mitchison, which is sort of a riff on systems biology. It's hilarious. Only a Brit could have written it. He makes allusion to this meeting in 1966 for which you see a picture on the right. At that meeting, if you look at the names, there was more brain power in that room, in that meeting, than anything I have ever been to. And you could ask, or it makes you reflect as to what will our ruminations look like 60 years in the future. Fortunately, we do not publish proceedings anymore, so no one will know. However, these proceedings are published, and I invite you to look at them. If you page through them, the only guy to come out well is Waddington. He knew what he didn't know, which is important, to know what you don't know. And everybody else is gobbledygook, by and large. But he was really on target. He knew his limits, and he knew where the gray areas were and everything else. So he comes across very well. As you could see by his placement in the picture, you could tell who the chief banana was. Okay, that is it. Thank you.
A quick question. There are two women in this picture. Do you know who they are?
Yes, that I do. It's said in the caption. I did not recognize the names. The people I recognized, which is of course my prejudice, one of them is Sanger, and I don't know. I didn't register. Anyway, it's in the caption of the original article, and you obviously can see it's a group of dead white people, mostly male. It was long, but I heartily recommend looking at the proceedings because you speculate what our discussions will look like in 60 years, and you recognize what was possible to foresee 60 years ago and what was not. It's very revealing. And René Thom was I wouldn't say Albert's rocker, but engaged in gobbledygook. He complained about the plethora of data in 1966.
Could you comment on why the notions that are human about robustness and development will map onto the mathematical properties?
Okay, that is an interesting subject that is not original with me. There is a book from late 90s that explores those parallels which are very good. So an embryologist would tell you that you perturb embryonic development is very robust. And what that means, for example, if you do xenopus, you could fertilize xenopus eggs in a petri dish. You squeeze the mother and get the eggs out, and then you take the testicles from the father, you chop them up, and you mix them in the petri dish, and you will get with high fidelity tadpoles. That is some demonstration of robustness, and it's common. This is the way frog labs used to run. So I tell that story to alpha males often, that they were basically used just for their testicles, which were frozen in the refrigerator. So one believes that's an example of robustness. Similarly for for developing mother for mammals, again, a lot of insults go on.
And so why does that give Morse Smale?
Right, and if you look at what really cuts the most important assumption of Morse Smale is structural stability, which means that if you take, what that means is that nearby parameters, you take all your parameters in the Michaelis-Menten, and that means in an open neighborhood of parameter space, you could take all those flows and diffeomorphically map them back to the original system. So it's a very strong assumption. It excludes chaos. Of course, if we were chaotic, we wouldn't be here talking about it, obviously. But it's a very potent assumption. And of course, then the other parts of the analogy are obviously, the Waddington valleys are fixed points or attractors, et cetera, et cetera. So there is that parallelism is actually quite old.
When you're fitting the potential in the neural tube, you also fit a metric. I saw you put it in there.
We don't need much of one, so it's a number. It's a diagonal. It's trivial. There are cases where you could imagine using it, and it would break the symmetry of the cross derivatives in the potential.
There's one question there. Yeah.
Yes.
Hi. I don't think I understand everything you
Yes.
talk, but here you're trying to infer the geometry.
Yes.
But there are groups of thought trying to infer like the function, the space that kind of push the differentiation
Yes.
forward in time. Is there a mapping between those two? How should I think
Okay,
about
so
it?
you have to give me data
Yeah.
where you perturb the relevant genes, or you add the relevant morphogens, or you do something to expose the regulatory coupling, and then we will fit your data in principle, or Dylan will fit your data. So, you know, we're fitting data. So you have to tell us something about your system, and then we will say that. But the fact that, again, I underline most emphatically that the mathematics tells you that these bifurcations are living in a low dimensional space, like 1 or 2, and they don't live in 10 dimensions. So living in 1 or 2 means that there is compression in the normal direction and expansion in the bifurcation sense. And if you see that in data, that is a strong indication that the model is sort of sensible. And if you don't see it in data, well, then you worry about the whole program. But it's a quantitative statement. And of course, there are degrees of, but you have to put a number on the compression. So with the that stem cell system that I mentioned ever so briefly in PNAS, you know, we could calculate we could calculate the contraction onto the one-dimensional manifold. We could calculate at the saddle points that there's one unstable direction with a certain eigenvalue. There are multiple stable directions with positive eigenvalues, you're contracting. And at the stable states, there are all contracting stable eigenvalues. And that is inferred from the potential. So the potential is fit, and then you could smooth things and differentiate. So it's all quantitative. No ifs, ands, or buts. It's a tight procedure. And one, I should note, one is never trying to parameterize the manifold directly in 5 dimensions. One is using the actual data points to define where the manifold is. And obviously, where points are rare, you infer that the potential is high because things slide down. So the whole procedure is living on experimental data points. Nothing interpolated by hand. It's all living on the experimental data points. It's a tight procedure.
One last question before we break for unstructured paddleboard. Sorry, unstructured time.
So, Erica, would all the dynamics you have presented determined by the gradient of its potential?
No, right. So we have gotten away. I've spent a lot of time working on semitogenesis, for which you have a periodic, temporally periodic system, and also a spatial wave. And the transition is from this periodic system into a bistable system, which is the rostral-caudal polarity of the somites. And how that transition from periodic to bistable happens is a very interesting geometric question. And we have a list of ways for orbits to break down, right? There's a saddle node on the circle, a SNCC so-called. There is collision with an unstable orbit. There is a homoclinic bifurcation, et cetera, or Hopf bifurcation. And none of those look precisely right. but it's a very interesting question. So precisely, simultaneogenesis is very much on our mind, and that transition from periodic to bistable, which is experimentally very, you wouldn't be here standing up if your somites were not Roche-Color polarized, right? So that's essential biology, and it is not entirely clear of the geometry, but so we're working with an experimental group to try to nail this down. But it's the question makes you do interesting experiments in an interesting parameter, spatial regime, and it also has shown me at least the utility of spatial transcriptomics to resolve some of that.
Okay, there are no further questions. Let's all thank Eric first. Okay.